This video will focus on mutation, repair, and recombination. And we'll focus on some of the topics shown on the screen here. And let's actually jump right in with the second point there, namely the different types of point mutations and some of their consequences. So we're looking at a table here that illustrates the different types of mutations, specifically point mutations or single base pair changes that we can find. And let's first of all make the distinction between transition mutations and transversion mutations. So transition mutations, we're simply referring to any time you replace a purine with another purine or a pyrimidine with another pyrimidine. Transversion, on the other hand, refers to mutations whereby you replace a purine with a pyrimidine or vice versa. We can distinguish among different types of transition or transversion mutations and some of their consequences. So for example, if we have a mutation whereby we change a base but we don't change the amino acid, that's referred to as a synonymous or a silent mutation. Ultimately, there's no impact on the protein produced, again because we're producing the same amino acid sequence as we would normally find in the wild type. On the other hand, we can discuss and identify missense mutations. We have two missense mutations illustrated here. This is a situation whereby a base is replaced by a new base that ultimately leads to the formation of a new or different amino acid being produced in a protein. That may or may not have implications on the actual function of the protein in question. On the other hand, if there is a base change that leads to an early stop codon, like we see here in our table, that's simply referred to as a nonsense mutation. That, of course, can have a big implication because you're producing a protein that's shorter than it should be otherwise. Toward the bottom, we see indel or insertion deletion mutations. So anytime you insert or delete a base, that has a potential to induce a frame shift mutation that will essentially throw off the reading frame and potentially form a entirely new sequence of amino acids in the protein until a stop codon is reached. So again, this simply illustrates the different types of point mutations and their potential consequences in the proteins that are formed. It turns out that some mutations are spontaneous. They can occur within organisms by a variety of mechanisms. Let's just briefly illustrate some of those here. So it turns out that most spontaneous mutations are simply a result of abnormal base pairing. So for example, we can talk about tautomers. These refer to isomers with different arrangements of atoms and bonds, and these are fairly rare, but they do occur and they can be incorporated into DNA. And so we see a few of these illustrated toward the right here. So normal base pairing illustrated toward the upper portion of this figure. So C with G, T with A. That's what we normally see, and that's what we normally expect, right? Complementary base pairing. But we can have these tautomers produced that can lead to mismatch base pairing. So for example, we can have this amino form of cytosine binding with adenine. So C and A, right? That doesn't match up. Here we have an enol form of thymine binding with guanine. Again, that doesn't represent normal base pairing. Okay, so these tautomeric shifts, they can occur. And again, if you think about it, when they do occur, they simply represent examples of transition mutations because we're essentially substituting purines for purines or pyrimidines for pyrimidines. Another example of a phenomenon that occurs is referred to as slippage, and this occurs during DNA synthesis or DNA replication. So slippage is illustrated here. It comes into play when DNA polymerase makes a mistake during DNA synthesis. So imagine, toward the top of our figure, imagine during DNA synthesis, we have a template strand shown in blue. So imagine DNA polymerase coming along, inserting complementary bases. But what can happen, especially when we have repeat sequences? So for example, here we have trinucleotide repeats illustrated. So in this case, CAG, 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 CAG. That could be repeated numerous times. What can happen when we have these repeated sequences is that DNA polymerase can essentially sort of lose track of where it's at, if you will. So it can produce some complementary bases and then sort of slip and go back to where it was a few moments ago and repeat rejoin, repeat, creating complementary DNA bases until it's finished doing its job. But ultimately, that can lead to a situation like we see here, where we form multiple extra copies of a repeated sequence. So in this illustration, we have 1, 2, 3, 4 extra copies of the trinucleotide repeat. So ultimately 12 extra bases here. So what we're doing is essentially, in this instance, we're producing DNA that is now longer than it should be. It has more copies of that repeated sequence. Likewise, a similar situation can occur that can lead to deletions, or fewer copies of the trinucleotide repeats than there should be. So this can occur, and it can have big implications. What we see illustrated here is a table that illustrates various diseases that come about as a result of having typically extra copies of a repeated sequence. So for example, here's Huntington's disease. CAG happens to be the trinucleotide repeat in question. Typically, most people have between 6 to 35 copies of this CAG repeat. However, when slippage occurs in the coding region for a particular gene, individuals with the disease can have anywhere from 36 to 120 copies of that trinucleotide repeat. So again, slippage can have big implications in terms of formation of various diseases. In addition to mutations occurring not only during DNA replication, they can also occur after DNA has been produced. So we can talk about spontaneous lesions. For example, this concept of depurination. We're simply talking about a situation where a purine base, for example guanine, can be separated from the sugar molecule. The same thing can happen with pyridines as well, but it's not as common or frequent as depurination is. So if guanine gets split from the base, it's possible that the guanine will be reattached, no harm done, or it's also possible that a different purine base would be attached. So for example, adenine. So if something like that occurs, you can imagine the situation after several rounds of DNA replication, where we go from a GC base pairing sequence to potentially an AT, or an AT to a GC. Again, if you happen to have guanine removed from the sugar, replaced with an adenine, that's how you can end up with a GC to an AT transformation. Okay. Likewise, a similar situation referred to as deamination. So for example, we can have cytosine losing an amine group, in which case it becomes converted to uracil. So imagine if you initially have a CG base pair, if that cytosine is converted to uracil, uracil will form base pairs with adenine. Uracil will also be replaced after multiple rounds of DNA replication. So you end up going from a CG sequence to a TA sequence. Okay, again, a mutation caused by this change, this deamination, brought about by a change in the cytosine to begin with. There are also chemicals simply referred to as analogs or base analogs. These are very similar to bases, and they can actually be incorporated into DNA. So for example, 5-BU or 5-bromouracil. This happens to be an analog of thymine. And it can form an ionized tautomer that pairs with guanine. So if you initially have a TA base pair, if the T is replaced with 5-BU, that can form a base pairing with guanine. That's what you see here. Imagine after another round of DNA synthesis, the 5-BU will essentially be replaced by cytosine. So you end up going from a TA sequence to a CG sequence. Likewise, another example, 2-aminopurine or 2-AP. This happens to be an analog of adenine that can pair with thymine. However, it can also be protonated, and when it is, it forms a pairing with cytosine. So if we have an AT base sequence to begin with, if we then replace the adenine with 2-AP, its protonated form, again, that can form a pairing with cytosine. So ultimately, we end up with a GC base pair, again, after several rounds of DNA replication. So again, going from an AT to a GC. Other chemicals can serve as intercalating agents. So for example, we're just talking about chemicals that have a very flat or planar structure to them. They can literally insert themselves between the base pairs of DNA. Often we take advantage of some of these. So for example, lithium bromide is often used to stain DNA gels. Lithium bromide can insert itself right into DNA, and it's also fluorescent. So if you shine UV light on it, it can fluoresce anytime it's binding with DNA. Radiation, of course, can also cause problems. So for example, think about ultraviolet radiation, right, causing potential problems with skin cancer. What it can do is literally interact with DNA and cause bonds to form between adjacent thymines. So these thymine dimers can be produced, and they can produce kinks in DNA that can actually change the structure of DNA, and that can cause big problems if they go uncorrected. Ionizing radiation, it too can interact with DNA and change the nature of DNA. It can literally change the structure of some of the bases within our DNA. And of course, that can lead to potentially big problems as well. So think about all these ways that mutations can occur. It turns out that there are a variety of ways that mutations are not only prevented, but also repaired if they do occur. So on one hand, think about DNA polymerase producing new DNA. Often it will, by mistake, it will insert the incorrect base. When it does so, it has the ability to detect that mistake, pause, insert the correct base, and when that occurs, of course, no harm done, right? It essentially prevented that mutation from being propagated in the new DNA that was being synthesized. On the other hand, some mutations, they can occur, right, and they can persist, and often they can be repaired. And there are a variety of ways that mutations can be repaired. We'll just briefly mention one example here, namely base excision repair. That's illustrated graphically toward the right here. So imagine a situation where you have a damaged base, illustrated in blue here. A variety of proteins and enzymes come into play essentially to excise or cut out that base and replace it with the proper, normally functioning base. So for example, an enzyme DNA glycosylase, it recognizes and removes the damaged base. When that occurs, we now have a site that's missing a base. So we say that's an AP site, an apurinic, or it could be an apyrimidinic site. So basically, right, we're talking about a situation where we're missing a base in the DNA. Another enzyme, AP endonuclease, comes along and cuts or nicks the DNA at that point. Then the oxyribophosphodiesterase comes along and removes a section where the nick was made previously. Then, essentially, we just have normal DNA replication. So DNA polymerase comes in, inserts the appropriate complementary bases shown in red, and then ultimately DNA ligase connects the bonds between adjacent nucleotides, thus sealing the nick in the DNA. So this is essentially a mechanism by which a damaged base can be removed and replaced with a normal base. And let's wrap this chapter up with a brief discussion of the connection between mutations and cancer. Cancer mutations can be categorized in various ways. One example are so-called oncogenes. When we're talking about oncogenes, we're talking about genes that play a role in cell division, and we normally naturally need lots of genes to be expressed for cells to divide. So we say they begin as proto-oncogenes. So proto-oncogenes are simply genes involved in cell division, but the problem is when those genes become mutated that lead to cancer, we say then they become oncogenes, right? So again, we're talking about mutations that lead to uncontrolled cell division. One widely studied example is the RAS oncogene. RAS comes from rat sarcoma. That's what it's named after. And it goes something like this. So within cells of many organisms, there are receptors. They can bind to growth factors. When growth factors bind to the receptor, that then triggers a change in a G protein known as the RAS G protein. When that RAS G protein is activated, that's what's illustrated toward the bottom of this figure, toward the right here. When the RAS protein is activated, that then triggers a whole cascade of events inside the cell, ultimately that leads to the production of transcription factors that lead to mitosis. Normally, after the RAS G protein is activated and activates the formation of transcription factors. It's then deactivated by binding GDP, guanine diphosphate, instead of GTP. So it binds GTP in its active state, then it becomes in its inactive state, again by giving up the GTP and binding GDP instead. What happens when individuals have mutations in the gene that codes for the RAS G protein, the RAS G protein is stuck in its active state. That's what's illustrated here. So essentially we have this protein, this G protein, always triggering cell division, even when it shouldn't. So again, this is just one example of an oncogene, right? A mutation in this gene that's causing uncontrolled cell division. On the other hand, there are other genes involved in cancer that are referred to as tumor suppressor genes. So tumor suppressor genes, these code for tumor suppressor proteins, which are constantly produced, and what they do is have the ability to detect precancerous cells and either help those cells become repaired, or they can trigger those cells to die. So they can trigger apoptosis or programmed cell death. And so these are a good thing. These are proteins that, again, help prevent tumors from forming, for the most part, in individuals. But of course, a problem comes into play when the genes that code for tumor suppressors are mutated and they no longer produce functional tumor suppressor proteins. So just as one example, P53, this is a gene in the P53 protein is of course, a tumor suppressor protein. It plays a big role in recognizing cancerous cells and causing those cells to destruct. If it turns out that P53 is associated with about half of all the cancers that are out there. So again, it plays a huge role in many, many cancers. So that concludes our brief discussion of mutations.